Okay, what's up, AP Bio? I'm here to just make a little video to explain signal transduction pathway in terms of T cells and their interactions with B cells. So what goes on first is that a fragmented antigen is shown, and it basically what happens is the B cell takes it in, and this is already a fragmentation of the antigen, and what happens is it goes through the lysosomes, where the lysosomes break it down to a peptide. Okay, so the peptide then binds to this MHC molecule, and so what this does is pretty much it moves the peptide onto the surface of the molecule. I mean of the cell, of the B cell, and this is pretty much our cell-to-cell interaction. And what this is pretty much, this is the site of where our transduction occurs. Okay, so what we have here is a better look at that protein complex. So this is our TCR, so that's a T cell receptor, and this is where the signal transduction occurs. This big bubble. And so let me break this down. So here we have a bunch of proteins, and what's going on is that these proteins are all pretty much going into a chain reaction of phosphorylating each other. So this is called a phosphorylation cascade. And so what's happening here, it's pretty much a successive chain reaction of kinase proteins that phosphorylate each other. And what the phosphorylation does is it pretty much activates each protein, and it tells them to phosphorylate another. So it's like pretty much dominoes. So once the first one is phosphorylated, they just constantly phosphorylate each other, and they activate each other. And by doing this, they pretty much come and make a ligand. So this is our ligand IP3, which is also a kinase protein. Okay, so what's going on here is that our ligand IP3, or which is a kinase protein, it's gonna travel to the ER. Right here, you can barely see it, but ER, our endoplasmic reticulum, and there's a certain ion-gated channel. Right? And that's right here. And so what IP3 does is IP3 binds to a certain ion in the ion-gated channel, which then produces the ion this ion right here, and you can barely see it, but it's calcium. Oh, it's what? What is this word? It's calcium 2 plus. Okay, so once our ion calcium 2 plus is released into the cytoplasmic environment, it goes through like a series of reactions, which results in the production of NFAT, which is our nuclear transcripting protein. Okay, so what NFAT does is it infiltrates the nucleus of the cell, and it binds to the DNA. And this binding is pretty much going to initiate the transcription of a specific gene. And when that gene is transcribed and translated, the actions committed by this new protein are going to allow for T cells to differentiate into specific T cells. So in this case, we need more helper T cells to deal with the antigen. Okay, so yeah, that was just an example of how signal transduction pathways work. Just a closer look, and to make it less complicated.